Molecular design and protein engineering are rapidly advancing fields with major implications for therapeutics, proteomics, and materials science. We'll explore innovative approaches to creating miniature proteins with high binding affinity and specificity. These engineered proteins have potential applications in drug discovery, biosensing, and advanced materials. Novel polymerization techniques within nanoporous materials also transforming these domains, enabling the development of new functional materials. The dramatic molecular structure visualization on the slide highlights the complexity and beauty of these molecular systems. It's a testament to the incredible advances happening in this field. These cutting-edge developments open up exciting new possibilities across a range of important scientific and technological areas. The research focused on thermally induced polymerization within nanoporous materials, creating novel composite materials with unique properties. The experimental approach involved heating N2 purged samples in a Kugelrohr apparatus at 170 degrees Celsius for 2 hours to induce the polymerization. Comprehensive characterization techniques were used to analyze the materials, including absorption and fluorescence spectroscopy, infrared spectroscopy, X-ray diffraction, and high-resolution transmission electron microscopy. Thermogravimetric analysis was conducted at a heating rate of 10 degrees Celsius per minute to evaluate the thermal stability characteristics of the materials, which is essential for potential applications. Mesoporous materials have emerged as powerful templates for creating novel polymer structures, building on pioneering work in the field. Researchers have demonstrated the ability to incorporate conductive polymers within the channels of these materials, as well as explore the photophysical properties of confined polymers. Recent innovations have advanced the field, with new approaches to polymer confinement and organization within the nanoporous structures. This has expanded the potential applications of these materials, including in areas like optoelectronics, sensing, and advanced materials development. The key focus of this presentation is on the process of incorporating polymers into mesoporous materials, a class of nanoporous solids with well-defined pore structures. We'll walk through the 4 main steps involved: synthesis of the host mesoporous silica materials, introduction of the monomer precursors, thermal polymerization within the confined pore spaces, and finally extraction and analysis of the resulting polymer materials. The controlled pore sizes and geometries of the mesoporous silica templates allow us to direct the formation and properties of the embedded polymers in unique ways. Careful control over each stage of the process is critical to producing well-defined polymer nanostructures with tailored characteristics. I'll highlight the key techniques and considerations at each step to give you a comprehensive understanding of this powerful approach to polymer synthesis and confinement. The research focused on analyzing the spectroscopic properties of polymers confined within nanoporous materials. Absorption spectroscopy measurements in reflection mode revealed unique optical characteristics of the confined polymers, with spectral shifts compared to bulk polymers. This indicated successful confinement and altered electronic structures of the polymers within the nanochannels. Fluorescence analysis using a specialized spectrophotometer setup showed emission properties of the confined polymers. Fluorescence microscopy provided visual confirmation of the polymer formation and distribution within the host material. Infrared spectroscopy analysis at KBR pellets samples confirmed the chemical structures and interactions between the polymer chains and the silica framework. The characteristic absorption bands provided evidence of successful polymerization within the confined spaces. X-ray diffraction patterns revealed the structural organization of polymers within the mesoporous framework. Optical microscopy provided visual evidence of polymer formation and distribution throughout the host material. Transmission electron microscopy allowed direct visualization of the polymer chains within the nanoporous channels at near atomic resolution. These complementary characterization techniques provided a comprehensive understanding of the structural and morphological features of the nanoporous polymer-based material. Thermal analysis is a key technique for understanding the thermal stability and decomposition behavior of materials. We conducted thermogravimetric analysis, TGA, on our samples using a Shimadzu TGA-50 instrument. TGA allows us to measure weight changes in the sample as it is heated at a controlled rate, providing insights into thermal stability. The samples were heated at a rate of 10 degrees Celsius per minute to fully characterize their decomposition profiles. To extract the polymer component, we suspended the composite powder in hydrofluoric acid, 55% concentration, at room temperature for 1 hour. This allowed the silica framework to dissolve, leaving behind the polymer. Finally, we bubbled nitrogen gas through the resulting clear solution to flush out any volatile fractions that were released. Mesoporous materials have been a rapidly evolving field since the pioneering work of Beck et al. And Korma in the 1990s. Significant contributions from researchers like Wu, Bain, Cardan, Nguyen, and Kageyama have expanded our understanding of polymer confinement and organization within nanopores. These advancements have led to new applications in optoelectronics, sensing, and advanced materials. The slide highlights some key references that have shaped the development of this important area of materials science. Protein grafting is a powerful approach for designing miniature proteins with high binding affinity and specificity. The key is identifying important alpha helical residues from natural proteins and grafting them onto small, stable protein scaffolds like avian pancreatic polypeptide, AP. This allows the miniature proteins to maintain the binding properties of the original protein, while offering advantages in stability, size, and potential therapeutic applications. Miniature proteins designed this way have high specificity, nanomolar affinity, and a stable alpha helical structure, all within a compact scaffold. This is an effective strategy for engineering small, highly functional proteins for various applications. BCL2 and BCLXL are central regulators of apoptosis, or programmed cell death. They have an anti-apoptotic function, meaning they inhibit cell death pathways. This is significant because BCL2 and BCLXL are overexpressed in 80% of B-cell lymphomas, helping cancer cells avoid apoptosis. Inhibiting BCL2 and BCLXL could be a therapeutic target, as it could restore apoptosis in cancer cells. These proteins bind to the BH3 domains of pro-apoptotic proteins, sequestering them and preventing cell death. The BH3 domain of the BAC protein is a 16 amino acid sequence that functions as a death agonist, meaning it can induce apoptosis or programmed cell death. This BH3 domain binds to anti-apoptotic proteins like BCL2 and BCLXL, which normally inhibit apoptosis. By binding to these proteins, the BAC BH3 domain can trigger the apoptotic pathway. When the BAC BH3 domain binds to BCLXL, it forms a 3-turn alpha helix that fits into a hydrophobic cleft on the surface of BCLXL. The BAC BH3 domain binds to BCLXL with higher affinity, CADE equals 0.35 μm, compared to its binding to BCL2, CADE equals 4.9 μm. This difference in binding affinity is about 1.4 kilocalories per mole, reflecting subtle variations in the binding pockets of these two anti-apoptotic proteins. The ability of the BAC BH3 domain to bind and inhibit anti-apoptotic proteins is a key mechanism by which it can induce apoptosis in both cell-free and cellular systems. The slide highlights the structural homology between the anti-apoptotic proteins BCL2 and BCLXL. Despite their high structural similarity, with a backbone RMSD of only 1.91A, there are some notable differences between the two proteins. 9 out of the 10 BCLXL residues that contact the pro-apoptotic protein BAC are also found in BCL2. However, there are 3 key differences in the primary sequence of the binding pocket between BCL2 and BCLXL. These include the conservative mutation of L112 in BCLXL to M115 in BCL2, the mutation of A108 in BCLXL to D111 in BCL2, and the mutation of S126 in BCLXL to R129 in BCL2. These changes alter the charge distribution in the binding pocket, which could impact the binding affinities and specificities of BCL2 and BCLXL. The goal of this project is to design a miniature protein that can target the BCL2 protein and disrupt its function. To do this, we analyzed the sequence alignment between the BAC peptide, which binds to BCLXL, and the alpha helical region of the AP scaffold protein. We looked at all possible alignments to find the optimal positioning of key residues, scoring each one based on conflicts between the AP structure and the BCLXL binding site. Next, we created models of each potential miniature protein bound to BCLXL to evaluate steric clashes and calculate the overlap between the van der Waals surfaces. Finally, we calculated the root mean square deviation, RMSD, of each alignment to determine how closely the miniature protein would mimic the natural BAC-BH3 domain structure when bound to BCL-XL. This analysis allowed us to identify the best candidate miniature protein design to target BCL2. The goal of this project was to create a protein that is well folded and has high affinity for BCL-XL, a key protein involved in regulating apoptosis. To achieve this, we analyzed different alignments between the alpha-parvalbumin (AP) protein and the BAC protein, which binds to BCL-XL. We scored the alignments based on conflicts between residues important for AP structure, shown in blue, and those critical for BCL-XL binding, shown in red. Alignments with multiple conflicts were disfavored, as they would likely produce proteins that were either well folded or had high affinity, but not both. The optimal alignment, alignment 3, positioned residues 2134 of AP with residues 7487 of BAC, minimizing conflicts and steric clashes while maintaining the critical binding interface. The final design preserved 6 BAC 7287 residues that contribute significantly to BCL-XL affinity, and 4 AP residues that comprise the AP hydrophobic core. The goal of this research was to design a library of miniature proteins and use phage display to select for binders to the target protein BCL2. The library was designed with 6 fixed BAC residues, 4 fixed AP residues, and 4 randomized positions. This allows for a diverse set of potential binders. The library was expressed on the surface of M13 phage, which enables selection based on binding affinity. 5 rounds of selection were performed, using GST-BCL2 immobilized on glutathione-coated plates. After the 5 rounds, 16 clones were sequenced, revealing 7 distinct sequences. This indicates the selection process was successful in identifying unique binders. The slide shows a table of selected miniature protein sequences that were designed to mimic the BAC-BH3 domain. The sequences are compared to the original BAC, 7287, peptide, showing the amino acid sequence, frequency of selection, and binding affinity, CADE, for each. Analysis of the selected sequences reveals a strong preference for amino acids similar to those in BAC and other BH3 domains. Surprisingly, 3 of the miniature proteins, PPBH31, PPBH32, PPBH33, contained a phenylalanine at position 28, which was not randomized in the original design, suggesting this mutation may have functional significance. The slide also includes a visualization showing the sequence alignment and highlighting the conserved and mutated positions compared to the original BAC peptide. This slide presents the results of a binding affinity analysis, comparing the interaction between BCL2 and different protein sequences. The key finding is that the miniature proteins PPBH32 and PPBH33 bind to BCL2 with much higher affinity than the BAC7287 sequence. Specifically, PPBH32 binds with 40-fold higher affinity, and PPBH33 binds with 60-fold higher affinity, compared to BAC7287. The CADE, or equilibrium dissociation constant, is a measure of binding strength. The lower the CADE, the stronger the binding. The BAC7287 sequence has a CADE of 4.9 μm, while PPBH31, PPBH32, and PPBH33 have CADE values of 52 nanometer, 80 nanometer, and 120 nanometer respectively. This data suggests the miniature PPBH3 proteins could be promising candidates for further development as potential therapeutic agents targeting the BCL2 protein. PPBH31 is a high-affinity binder of the BCL2 protein. It binds BCL2 with an equilibrium dissociation constant, CADE, of 52 nanometer. This is 100 times tighter binding than the natural BAC7287 peptide. The polyproline helix in PPBH31 contributes about 2.5 kilocalories per mole to the binding energy. This highlights the advantage of the pre-organized structure of PPBH31 compared to the more flexible BAC7287 peptide. The binding data and fitted curves shown in the graph illustrate these affinity differences. The slide presents structural analysis of the PPBH31 protein, including its circular dichroism, CD, spectrum and thermal stability. The CD spectrum shows that PPBH31 contains one or more alpha helices, with approximately 60% of the protein adopting an alpha helical conformation. The thermal stability analysis indicates that PPBH31 undergoes a cooperative melting transition with a melting temperature, trademark, of 65 degrees Celsius, suggesting it adopts a stable, well-folded structure. This is in contrast to the largely unstructured BAC7287 peptide, highlighting the structural differences between PPBH31 and this related peptide. The data suggests PPBH31 contains an extended, contiguous alpha helix, which is a key structural feature of the protein. The slide compares the structural properties and binding energetics of two different peptides, the unstructured BAC7287 peptide and the prefolded PPBH31 peptide. The BAC7287 peptide has little alpha helical content in solution, meaning it needs to undergo a structural change to adopt the alpha helical conformation required for binding. This structural change requires energy, which represents a binding energy cost that must be paid upon binding. In contrast, the PPBH31 peptide is already 60% alpha helical in solution, so it does not need to undergo this structural change. The lack of a folding energy cost for PPBH31 results in an approximately 2.5 kilocalories per mole advantage in binding affinity compared to the unstructured BAC7287 peptide. This structural basis explains the enhanced affinity observed for the prefolded PPBH31 peptide relative to the unstructured BAC peptide. The slide presents data on the binding specificity of the PPBH31 protein. It shows the dissociation constant, CADE, values for PPBH31 binding to several different protein targets. PPBH31 binds very tightly to BCL2 and BCLXL, with CADE values in the nanomolar range. In contrast, the CADE values for binding to protein kinase A, carbonic anhydrase 2, and calmodulin are much higher, in the 30,100 μm range. This large difference in binding affinity suggests that PPBH31 interacts very specifically with BCL2 and BCLXL, and does not bind promiscuously to other hydrophobic peptide-binding proteins. The high specificity of PPBH31 for BCL2 and BCLXL makes it a promising candidate for therapeutic targeting of these proteins. The slide compares the binding preferences of two different molecules, the natural BAC-BH3 peptide and the designed miniature protein PPBH31 for the proteins BCL2 and BCLXL. The BAC-BH3 peptide shows a 14-fold preference for binding to BCLXL over BCL2, with a binding affinity, CADE, of 0.35 μm for BCLXL and 4.9 μm for BCL2. The designed PPBH31 miniature protein maintains a similar level of discrimination, with a 7-fold preference for BCLXL, CADE equals 7 nanometer, over BCL2, CADE equals 52 nanometer. Despite operating at much lower concentrations, the prefolded recognition helix in PPBH31 is able to differentiate the molecular surfaces of BCL2 and BCLXL just as effectively as the natural BAC-BH3 peptide. Miniature proteins offer several key advantages as binding agents compared to traditional antibodies and small molecules. They have high specificity, allowing them to discriminate between closely related protein surfaces with precision. They can bind their target proteins at nanomolar affinity, which is physiologically relevant. Their compact size makes them small enough for cellular penetration and reduces their immunogenicity. Miniature proteins also have a well-folded, structurally stable design that resists proteolytic degradation. These properties make miniature proteins a promising class of binding agents for research and therapeutic applications. The phage display library, PPBH3LIB, was created by cloning a specific DNA sequence into a phage vector. This allowed the library to display a diverse set of peptides on the surface of the phage particles. M13 phage were then produced and purified using standard methods. The phage library was incubated with a mobilized human GSTBCL2 protein to allow binding. After washing to remove unbound phage, the bound phage were eluted and used to infect E. coli. This selection process was repeated over multiple rounds to enrich for phage displaying high-affinity peptides. All the peptides used in this study were synthesized using standard solid-phase methods, following well-established protocols to ensure efficient amino acid coupling and proper side chain protection. After synthesis, the crude peptides were purified to homogeneity using high-performance liquid chromatography. This allowed us to remove any truncated sequences or synthesis byproducts, ensuring we had the target peptides in high purity. MALDI-TOF mass spectrometry was used to confirm the correct molecular weight of each peptide, verifying that we had successfully synthesized the target peptide sequences. Some of the peptides were labeled with acetamidofluorescein, a fluorescent tag. This allowed us to use these peptides in fluorescence polarization binding assays, enabling quantitative measurement of their interactions with proteins. Fluorescence polarization is a powerful technique for studying molecular interactions. It allows us to measure the binding affinity between a fluorescently labeled molecule and its target. In this study, we used a specialized fluorimeter to perform these binding assays. The experiments were carried out at 4 degrees Celsius in phosphate-buffered saline, with some additional conditions for coolmodulin experiments. To generate the binding data, we incubated the fluorescent peptides with varying concentrations of the target proteins. We did multiple replicates to ensure the statistical significance of the results. Finally, we analyzed the binding data using appropriate equations to determine the equilibrium dissociation constants, or CADE values. The error bars on the data represent the standard error from at least 3 independent trials. Circular dichroism, CD, spectroscopy is a powerful technique for analyzing the secondary structure of proteins and peptides. We can use an AVIV 202 CD spectrometer to collect CD spectra for 2 peptides, PPBH31 and BAC7287, at 4 degrees Celsius in phosphate-buffered saline. The instrument was carefully calibrated to ensure accurate ellipticity measurements across the wavelength range of interest, which was 190,260 nanometers. Wavelength scans were performed to analyze the secondary structure content, focusing on the characteristic minima at 208 and 222 nanometers that indicate alpha helical structure. We conducted temperature-dependent measurements, monitoring the ellipticity at 222 nanometers as we gradually increased the temperature from 4 degrees Celsius to 90 degrees Celsius. This allowed the determination of the thermal melting temperature, trademark, of the peptides. To quantify the secondary structure, we calculated the mean residue ellipticity, theta MRE, from the raw ellipticity data, normalizing for factors like peptide concentration, path length, and number of amino acid residues. By comparing the theta MRE at 222 nanometers to reference values for 100% helical peptides, it is able to estimate the alpha helical content of the samples, providing a quantitative measure of their secondary structure. BCL2 is a protein that helps cancer cells avoid apoptosis, or programmed cell death. High levels of BCL2 are found in 80% of B-cell lymphomas and other cancers. BCL2 protects cancer cells from treatments like chemotherapy and radiation. Inhibiting BCL2 could make cancer cells more susceptible to these treatments. Miniature proteins like PPBH31 can bind to and inhibit BCL2 with high affinity and specificity. These miniature proteins may offer advantages over small molecule inhibitors in terms of targeting BCL2 effectively. Combining BCL2 inhibition with chemotherapy or radiation could be an effective cancer treatment strategy. Miniature proteins are a promising new class of therapeutic agents, but there is still more work to be done to fully realize their potential. One key area for future development is further evolution, running additional rounds of selection to enhance the specificity of these miniature proteins for their intended targets. Improving delivery methods to get the miniature protein into cells is also crucial, so we can effectively deliver them to where they need to exert their therapeutic effects. Enhancing the stability of the miniature proteins through engineering increased proteolytic resistance is vital for enabling in vivo applications. Finally, we want to explore applying the protein grafting approach we've used to other therapeutic protein targets, expanding the reach and impact of this exciting new technology. This research demonstrates the versatility of protein grafting and evolution for producing miniature protein ligands that can bind to protein surfaces with high affinity and specificity. The ability to design these compact, functional miniature proteins represents significant advances in both materials science and protein engineering. These miniature proteins have potential applications ranging from novel therapeutics to functional materials, as they can be used to dissect, modulate, or analyze specific protein functions within complex biological systems. Addressing the challenge of designing molecules that can bind protein surfaces with nanomolar affinity and high specificity opens new avenues for intervention in disease processes. In summary, this work showcases the therapeutic potential, research tools, and validated design methodology enabled by this approach to creating functional miniature proteins.